cycle. The cell cycle is a description of the events that happen after a cell divides, from the moment it's done dividing until it divides again or ceases to exist. And we can divide that into 3 stages. As I said during the lectures on Monday, the last 2 stages kind of overlap with one another, mitosis and cytokinesis. So interphase is all about growing and doing what you're supposed to do. Mitosis and cytokinesis are all about dividing. Mitosis, you're dividing the nucleus, and in cytokinesis, you're dividing the rest of the cytoplasm. So interphase is always a very active period in the cell, and for that reason, the DNA is in the form of chromatin. It's being used, it's being read. The cell continues to grow till it reaches the limits of growth, and if it's going to divide, it will replicate its DNA. That means to make a copy of its DNA from start to finish in preparation for mitosis. You've also got to make copies of other things as well, like organelles, membranes. You're going to need more membrane, and some other chemicals that you need as you go from being 1 cell to 2 cells. The phases of interphase include S, in which DNA is replicated. S is preceded by G1, which is a normal growth phase. And then if a cell is going to divide, it will go through S and then G2, which is they are both gap phases or growth phases, where the structures besides DNA are going to be replicated as in order to have enough to make 2 cells out of 1 cell. Mitosis, or the mitotic part of cell division, is the actual production of 2 daughter cells from an original single cell, and that would include mitosis, where the nucleus is divided, and that will overlap to some extent at the end with cytokinesis, where the cytoplasm is divided. So mitosis is divided into prophase, in which the chromatin, which is not going to be used during this process, is condensed and packaged up into the form of chromosomes that are more easily moved. The nuclear envelope is going to disappear, and the materials are going to get organized so that they can be equally split between the 2 daughter cells. Part of that process will move them towards the middle of the cell in what we call metaphase. And in metaphase, those chromosomes will be aligned on the metaphase plate, an equatorial plane, somewhere in the middle of the cell. Be 2 sister chromatids joined by a centromere. And at the end of metaphase, the centromeres will split. The chromatids will begin to move away from each other, and we'll be in anaphase. And as in anaphase, they will move to opposite sides of the cell. Eventually, they will get far enough apart that we will call it telophase. And in telophase, the chromosomes will go back to chromatin, the nuclear membrane will reform, the nucleoli will become visible again, and for a brief time you'll have 2 nuclei in 1 single cell, but that single cell will be in the process of going through cytokinesis. A cleavage furrow will form, and it will begin to split into 2 cells. So we got prophase, metaphase, anaphase, and telophase. Preparation, lining in the middle, chromatids moving away from each other, and 2 nuclei forming in the cell. And we can look at it here in more detail. You will look at some of these images in the lab on Thursday. Cytokinesis is the process of dividing the cytoplasm, and that happens very late in the mitotic part. It really overlaps with late anaphase, early telophase. And the contractile ring of proteins will form the structure here, which initially is called the cleavage furrow. That cleavage furrow will continue to twist and shrink until you get a smaller and smaller point, and eventually those 2 edges will separate from one another to give you 2 genetically identical cells from the first cell. That is going to be very strictly regulated. It depends on the cell type. Some are better at it than others. Some do not divide. Things like skin cells, intestinal cells, and our blood-forming cells are constantly dividing. The end of each one of those chromosomes has a structure on it called a telomere, and the length of that telomere is pretty predictive of how many times a cell has divided. So it's pretty good evidence that the shortening of telomeres is contributing at least to age and contributing to the number of times that a cell can divide. Hormones are going to be controlling that. Contact inhibition, as I talked about on Monday, is going to be part of that. Cells that are in touch with other cells don't tend to divide. And when we lose control of mitosis, we get a disease called cancer. We can get a disease called cancer, but if we lose control of cell division, we do develop tumors. Tumors can be benign, in which they are local, not moving around, not particularly dangerous, but they can continue to mutate and they can become malignant. Once they become malignant, they become invasive. They leave the initial site and go to other sites, and that is called metastases. There are genes we know that are associated with cancer. They are normal genes that, when mutated, cause cancer. And it takes a lot of mutations accumulated over a lifetime to get most cancers. Oncogenes are abnormal forms of a gene that controls the cell cycle. I'm not sure I agree with that. Oncogenes are regular genes that control the cell cycle. When they become mutated and we lose control of the cell cycle, they become associated with cancer. Sometimes that's because they're overexpressed. Sometimes it's because they're always turned on when they should be regulated. There are also tumor suppressor genes, which are normally limiting mitosis, but when we lose those, when they become inactive or removed completely, we lose the ability to get the mitosis under control. So you can think of oncogenes kind of as an accelerator in the cell, and tumor suppressor genes as kind of a brake in the cell. So if your accelerator sticks and your brakes fail, you're going to be in trouble. So what we get in development of cancers requires a number of mutations, usually over the course of a lifetime. It also has environmental exposures involved, but at some point a healthy specialized cell begins to divide and does not divide with control. As mutations accumulate in that in the cells that result from that healthy cell, you become a tumor, localized growth, and then that localized growth will accumulate more mutations, and as it accumulates more mutations, that can lead to invasion of local tissues and eventually metastasis, where it moves to other locations. Okay, and that will do it for this section.